Thank you, Ben, and again, thank you for putting this together. I'm sure the audience has enjoyed it so far, and kudos to you for finding a way to still get good science out there. And as Ben said correctly, the work I'm going to be talking about is something that just recently got published, and unfortunately, in keeping in the theme of open science, the paper is an open access journal manuscript. And so when you see, I'll have the citation in the presentation, and you can go on to that link and get it. And it's available now for you to, wherever you are, you can get it for free. So it's published through the Electrochemical Society, which is a society that's very, very into making sure that science is open and available for everyone, no matter where they are. So just a quick introduction of who I am, since you can't really see me. I'm Damilola, Daramola, I go by Dami for short, born and raised in Nigeria. And actually I wanted to also highlight that, just like a lot of countries currently, right, especially what happened in the summer, Nigeria is also going through, the citizens are demanding and requesting good governance. And so you've probably heard of stuff going on in the news. And again, like I said, similar to what happened here in the US in the summer and worldwide, citizens are finding a way to request good governance, and I wanted to just highlight that. And then I came to the US in 2000, got my education at Ohio University, did a number of things, including working with a chemical company called Ashland Chemical, and then came back to Ohio University. And as of this past fall, I became an assistant professor in the chemical and biomedical engineering department. My interests are obviously in electrochemical wastewater remediation and computational electrocatalysis, which is what I did for my dissertation, and then polymer composites as well. And so, as Ben correctly said, this talk is going to really be about electrochemical wastewater remediation. And really, when we talk about wastewater remediation, we're looking at the approach of handling nutrients, specifically nitrogen and phosphorus. And so I will just, again, give a quick introduction as to what, in general, how these, what these nutrients are, what their impact is, and some of the different ways in which you can handle them. And I'm not sure what the current audience is. I'm not sure what your level of familiarity is with electrochemistry. So I'd like to talk a little bit about electrochemistry itself, just very brief, and then go into how we can leverage electrochemical techniques for wastewater remediation, and then the methodology for this work. A little bit is theoretical to support experimental work that was performed, and then I'll introduce to some of the people that I work with. And so globally, and whether you're in the US or like Nigeria, where I'm from, we understand that there's a famous line that by 2050, right, the global population is expected to be around 9 million. And associated with that is the fact that you have a lot of urbanization, where you have people moving from a lot more migration from rural regions to urban regions, where you have these huge population centers. I'll give an example. Like I grew up in Lagos, Nigeria, and Lagos is a really small state, but it has one of the largest populations in Nigeria, just because there's been a lot of movement from rural areas to these population centers. And if you combine the fact that you have population growth with urbanization, population growth means obviously we need to have more food to feed people, and that overlaps with the fact that to grow food or to generate food, you need water, right, to sustain that food, and food production is also an energy intensive process as well, right? So these three things overlap in what is known as the food, energy, water nexus or FEW. And the systems there overlap in ways that also have an impact on it and an impact on the environment. So I'm going to play this short video that talks a little bit about some of that impact, and then go from there.
What is eutrophication? It's a problem that should matter to you, whether you live near the ocean or not. That's because it begins wherever people live and ends with damage to resources we all use and enjoy. It all starts when nutrients get into lakes and oceans. Remember, what's waste to humans can be food to plants and other creatures. Nutrients feed algae like they do other plants. Algae grows and blocks sunlight. Plants die without sunlight. Eventually, the algae dies too. Bacteria digest the dead plants, using up remaining oxygen and giving off carbon dioxide. If they can't swim away, fish and other wildlife become unhealthy or die without oxygen. But it doesn't have to be this way. Protecting marine resources starts with sound agricultural and waste management practices.
Right. So if we think about the ways in which these urban centers operate, right? So you have a lot of people living in a small condensed space. There's a more intense food consumption, right? Which means you also need to have more intense food production. And as we eat and consume, our body isn't that efficient in converting the food. So we have waste that we generate. And also these, if you think about these concentrated food production facilities as well, whether it's animals or plants, there is also a huge amount of waste generated there as well. And that is what is part of this excess nutrients that cause an impact on the environment overall. So if we, one of the ways in which concentrated or intense food production occurs is in facilities known as concentrated animal feeding operations here in the United States. And so you have this livestock, either cattle or poultry or swine, and they are in these huge facilities. And to handle the waste, the facilities use what are known as lagoons. And so you can see some structures here in which the waste from these structures end up going into this lagoon. And so now these concentrated animal feeding operations also have land as well. And so the way they handle all of this waste is to pump out, because the waste itself, as I mentioned, right, your body, and same thing with animals, you consume food, you consume nutrients, but you don't convert it efficiently. And so there's excess nutrients in your waste. And these nutrients are things that plants can use. And so this lagoons, the water in there, the manure in there can be used for crop production as well as part of the supply chain. And so that is part of what leads to that eutrophication that was mentioned in that video. You have the fertilizer, the excess either leaching through the soil, or when it rains through erosion and things like that, it runs off into the water. And it's well known that typically when fertilizer is applied, it's not applied typically in the amount that the crop needs, is applied based on yield, in the sense that rather than applying it saying, okay, well, the corn needs X amount of nitrogen, X amount of phosphorus, it's more so how much nitrogen or phosphorus can I add to the land to get X amount of crop afterwards. And so you have a lot of excess fertilizer applied. And so just like the plants feed on the fertilizer, the algae on the water feed on those nutrients as well, and they grow, and that's what ends up causing eutrophication. So the idea here is, can we find a way to take the waste in these lagoons, which can control nitrogen and phosphorus, and use a technology to do some conversion of those species, right? And typically, the approach here is to take an external driving force, which is magnesium, combined with nitrogen and phosphorus to create a product known as Truvite. So this itself is fertilizer as well, but the difference is this is more of a slow release fertilizer. So when it's applied, it doesn't release the nitrogen and the phosphorus right away. It releases it over time, which is a way to control this runoff that you get. The other thing is, once this Truvite is generated, that's something that can be sold. And then, depending on how efficient this process is, you can now send that manure back to that lagoon, and then that can now be sprayed on the land, and you essentially eliminate this bleaching and runoff because you've captured some of these nutrients in a solid form. And so that is one way to handle this issue. And as mentioned at the end of that video, the best way to handle eutrophication is by improved agricultural management processes. So this is one of the ways to do that. And I'm talking about animals here, but this also happens at municipal or human wastewater treatment facilities as well, where if you use a digester, you have excess nitrogen and phosphorus generated as well. The difference in the wastewater treatment plant is that they have technologies to handle that excess. But what ends up happening is you raise the amount of energy that you need to consume at those plants because of this excess nutrient that's generated by this digester. So again, you see that impact of food, energy, and water, where raising or treating the water itself before you send it out from a wastewater treatment plant also raises the amount of energy that wastewater treatment plant will consume. And so some of the ways that this nitrogen and phosphorus can be handled is by using chemicals or biology. And so if the approach is to generate struvite, like I showed in the previous picture, you would take a solution that has your nitrogen and phosphorus species, and usually these are in orthophosphate and ammonium forms, and you would add magnesium to that to generate this struvite solid that I showed you earlier. So that's a reaction that you can see on the bottom of the screen. You take your nitrogen species, your phosphorus species, combined with magnesium to generate this chemical known as struvite. Or you can, and again, if we use the human example, rather than using magnesium, iron-based salts or aluminum-based salts are used in the wastewater treatment plant to handle phosphorus. On the biology side, you have these biological reactors to do the consumption. So I'm showing you a picture here of a wastewater treatment plant, for instance, and you will see that typically when you have microbiological treatment of wastewater, you have these huge reactors that have to do that conversion just due to economies of scale. Now, if we're going to deploy this kind of technology or either of these technologies on the farm, for instance, number one, knowing how much magnesium to add requires some domain knowledge. So that is not something that's can be readily or how would I say user-friendly for the farmer in this case. And a lot of this work currently is being deployed in Europe, so not a lot of it is being used here in the US. The other thing to consider is, if you look at the size of these facilities, a facility like this is a centralized location. On the other hand, I'll use Ohio as an example. If I want to, these are the different concentrated animal feeding operations in the state of Ohio, right? And so a centralized facility would work for a region right here where you have a lot of the farms really close together, right? And so you don't have that far to travel in terms of transporting the waste to treat it. But if you look out on the eastern part of Ohio, or maybe a little bit more central, you have these farms dispersed, right? And so a centralized location like this would not quite work for the economies of scale that you require. And so an alternative to that is to use a technology that is modular in the sense that it's something that you can either scale up or scale down depending on the actual application that you need, something that is easy to operate or control. And a key thing to any technology development is that it should be cost competitive, right? Cost competitive in terms of what is currently being used, but also if it's cheaper to just take your waste and transport it off site, then that is the technology you're going to use. You're going to do transportation and just transport that issue somewhere else as opposed to trying to handle it. And so that is where electrochemistry or electrochemical technologies come into play. Electrochemical technologies are considered modular in the sense that they don't require economies of scale to be economically feasible, right? They don't have to be huge or humongous. You can scale them according to the specific application required. They're also considered what you would call a process intensification platform. So in chemical engineering, we have what we call these different unit operations. So you might have a reactor that does conversion. You might have a separator that once you've done your reaction, you want to separate your product of interest. You want to separate that out from everything else. And so if you have a technology or a setup that can do both of those things simultaneously, do a reactor, a reaction and separation simultaneously, you would refer to that as intensifying your process, because you can do multiple unit operations or multiple operations in a single form. And so this is an example of an electrochemical technology. It's a fuel cell. You have hydrogen coming in and oxygen coming in to combine to form water. But in that conversion, you also have electrons being generated, and those electrons can power an external appliance. And so what you have in this system is you have a reaction going on, right? You have oxygen, hydrogen, and oxygen sort of reacting together. So you have a reaction, and then you have this, what you call an electrolyte, which is a membrane that separates your reactants from each other. So you have separation and reaction sort of happening in that particular instance. And there's other ways in which electrochemistry is considered as process intensification, but that will be an example of one. The other thing is in an electrochemical technology, you're either controlling the voltage, right, or controlling the current. So it's and most of these you do by just flipping a switch or something. And so in that regard, these technologies usually have integrated process control as well. So in that way, they are, like I said, user friendly, and the fact that they're modular provide advantages in comparison to current comparative technologies that I mentioned before. Some examples of electrochemical technologies, we have batteries, right, which we're all familiar with. You're taking the chemical energy in the battery and converting to electrical energy to power an appliance. And then you have something like this, which is a fuel cell. So I usually tell students a fuel cell is like a battery that just requires constant chemicals being pumped into it. So it does the same thing. It takes chemical energy and converts it to electrical energy. And then on the other side, you have what you call electrolyzers, which take electrical energy and make them into chemicals or products. And that is really what this technology I'm going to be talking about centers around, where we're taking electricity and using it to drive or to recover or to reduce the nitrogen and phosphorus that I mentioned earlier to make this struvite. And so the main thing to remember or to recall is that in your electrochemical system, you have two main parts. You have what you call an anode and you have what you call a cathode. And these two electrodes or positions, I would say, in your reactor are where the reactions happen. And I'm going to be talking about this handling of excess nutrients within the context of phosphorus, although the key thing to remember is that nitrogen and phosphorus are the main drivers for eutrophication. So there is some, I would say, disagreements of whether phosphorus is worse or nitrogen is worse. But if you are in an inland like Ohio is, where you are bordered by a lake, for instance, it's being proposed that phosphorus is the main issue there in those waterways. But if you're in a coastal region, so say Florida, for instance, or perhaps California, then you have the conversation has been that nitrogen is more of an impact there. But really, these two are coupled together. They have an impact there. So even though I'm talking about this in the context of phosphorus mainly, this technology can capture some nitrogen as well. So in our system, what we have is we have your phosphorus species, which can be, they have different forms depending on the pH of the system. So if you have an acidic solution, you have something more of this form, which is H3PO4. If you have more base in the system, you're going to have just a PO4 form, right? Because as you can see, as I add hydroxides into the system, I drive my equation from an H3PO4 to like other forms that don't have the hydrogen in it. And so in this system at the cathode, we have two reactions that can occur depending on what the potential that's applied. It can be oxygen reduction, so you're taking oxygen and forming hydroxides there, or it can be water reduction, where you're taking water and forming hydrogen and hydroxides. And so the way this technology is working is that it is making the conditions possible for you to have this high pH. And this high pH drives the formation of the phosphate species, which are required for struvite deposition. So it's sort of an indirect process where you take your system of oxygen and water and you make your hydroxide at the cathode in this case. And then those other hydroxide ions convert these phosphate species to just PO4, and then that is how you get your struvite to form. And so the initial work that was done before I joined the group was to understand the different parameters that affect the process. And so this is a graph that shows what you call the residual current. So where we're controlling the potential or the voltage in the system, right? Sort of like you're controlling the thermodynamics of what's happening at the surface. And then what you notice is that, at negative 0.6, the current stays high. At negative 0.8, the current starts to decrease. And then at negative 1.1, the current decreases significantly and goes to 0. And the difference is here is that, remember, what we're forming is a solid product. And so as I drive my reaction and I form OH, and the OH forms the phosphate and the phosphate forms the solid is going to deposit on the surface of this electrode. And so when you have deposition on the electrode, these reactions slow down, right? They don't have access to the surface of the electrode anymore. And so that is what accounts for this huge current reduction. So this is a good thing, right? Because what this tells us is that we're forming this solid that we want to form, and we're recovering phosphorus from the solution. So at this higher potentials, or at these less negative potentials, there is no struvite, or there's no solid deposition forming. But then as I drive it farther, I get more struvite deposition. Now, the other thing to keep in mind here is that what is going on is that if you are low, the lower your potential is, or the more negative we drive this value to, the more energy you're going to consume. And if we're talking about making something economically feasible, we have to keep in mind that we can't just apply a huge amount of energy. We want to make sure that we balance the amount of energy that we're applying to our system. It was also discovered that the flow conditions, so the turbulence in the system, has an effect on that struvite deposition as well. So again, as you can see, as I go so in that system, and I'll show you the configuration in a second, this was done in what is known as a rotating cylinder electrode system initially. And so when the system is static, when you have no turbulence in the system, you can see that my current just stays high, which means I'm not getting any struvite deposition on the surface of my electrode or my cathode. And then as we start to increase the turbulence in the system, you get more and more struvite formation. But then you also notice that if you go too high, you lose some of that struvite as well, because after a while, if your flow conditions are too high, rather than getting deposition, your flow is going to just basically remove the struvite that you form on the surface of the cathode. So there's a balance there as well. The other parameters that were discovered that affect the system are temperature and competing ions, because there are other ions that compete for the phosphorus, which means that rather than forming struvite, we're forming something else. So there's what you would call a selectivity issue that we have to be considerate of as well. So after these parameters were discovered, the goal then was to transition from a rotating cylinder configuration to an actual configuration where you can measure or quantify how much phosphorus you're actually removing in the system. And that was the experimental portion of this paper that I mentioned. So this citation here is something, like I said, it's readily available, it's open access. So if you note that or copy that or take that note down, you'll be able to find that and you can download it right away. And so in the experimental setup, now you see that we're noting this as a working electrode and a counter electrode, which is, and with electrochemical jargon, to say that the working electrode is where the reaction of interest is happening, and the counter electrode is just to balance, one is generating electrons, the other one is consuming electrons. So this is sort of to balance that. So in our case, the cathode is our working electrode because that's where the struvite is formed, and the anode is our counter electrode here. So in the experimental conditions that we're using, so we're trying to mimic a wastewater system. So we're using synthetic wastewater, and we use in the experiments, the magnesium, nitrogen, and phosphorus are combined in this ratio. Stainless steel was used. Stainless steel is not necessarily the best catalyst. It's not the most efficient in terms of converting oxygen to OH or water to OH, but it's cheap. And so thinking about that balance again between something that works and something that is economically feasible is something that's really important to chemical engineers. And so in this approach, we're going to use stainless steel to look at the reactions and then consider where we might want to operate to manage that economic feasibility. Because if your catalyst is expensive, it basically sort of kills the technology from the get-go, or it can, right? It's not always the case, but it can. And so so in this configuration, over time, as the phosphorus in this solution, the nitrogen in the solution is collected on the cathode, we can monitor the reduction of phosphorus in the solution, and that will give us a sense of how well we're recovering. And so usually in describing a technology, one of the values that we discuss are what we call figures of merit that compare how the reactor operates. So for instance, in reactor engineering, we have things like, okay, well, how well am I converting my species? How well am I, how selective is what I'm generating? So if I want product A and I'm getting product A and B, how much of A am I getting and how do I quantify that? So in this particular case, we're quantifying three things in addition to the currents that I showed you previously. We're quantifying how much phosphorus we're removing. So what's the efficiency of our phosphorus reduction? We're quantifying the current efficiency, which is basically a measurement of if I know how much electricity I'm sending to the system, I should know how much product I'm generating and how much of my electrons are going to my product and how much of them are going to something else. And so this is, in chemical engineering terms, it's sort of the selectivity measurement as well. But in this case, we're looking at how the electrons are being converted to products. And then we have the specific energy. This is the stuff that part of what you're going to be paying for to operate your system, right? In this case, it's an electrochemical technology, so what we're going to be paying for is electricity. And so that is something that's important to quantify as well. So if we're thinking just broad strokes, we want our phosphorus removal to be high, right? Because the goal here is to reduce the phosphorus in the solution so we can, again, reduce the impact of those nutrients downstream in, lakes and rivers and things like that. We want the current efficiency to be high, right? Because we want as much of our electricity to go to the products of interest, which in this case is true, right? And then want the specific energy consumption here to be low, okay? Because again, there's a cost associated with that. And usually here, the current efficiency is associated with the size of your system. So if your electrons are not going to products that well, then you would want to make your system bigger so that way you can make more products, basically. So this affects the size of your system. This affects the cost of operation. And so in this particular configuration, what you're seeing is and again, the details are in this paper, but what you're seeing is the effect of the potential we apply in this configuration on these different figures of merit. And so at an applied potential at the cathode of negative 0.8, we really get almost no struvite deposition. We get some because the current decreases, right? But we don't get as much as we would expect because we don't have the sharp drop off that everything else has. So we drop off in terms of how much electricity, how much current we start with, because that's a sign of how much we're depositing. And you can see those numbers here. We're removing some of the phosphorus, but not at a high percentage. So maximum over 100 minutes, we're removing about, let's say 30% or so of phosphorus, right? And negative 0.8, we can see the current efficiency there. So it starts off high and starts to decrease again, because once you start to cover the surface of the electrode, you're not that efficient in generating product as well. And you can also see it has a low energy consumption, but we want to look at all these parameters together. So low energy consumption, but low recovery is not really ideal. So if you look at negative 1.1, which was the voltage that was used for the rest of the analysis for these other systems, you can see that at negative 1.1 volts, we have almost 80-something% recovery of the phosphorus, and we're able to maintain 10% efficiency is not that great, but we haven't optimized the system yet. This is sort of a first pass. Well, at least we're able to keep a high enough efficiency over a long time in comparison to everything else. And then we can see there again, these are the upside down triangles, by the way. At negative 1.1, you can see that the energy consumption is low. On the other hand, at negative 1.5, so at around negative 1.1, we have mostly oxygen reduction happening, but we might get some hydrogen formation as well. As we drive the potential to more negative values, you're going to get more and more of this hydrogen generation in your system. And so what we're seeing here is that at negative 1.5, we still get struvite formation. Why? Because, well, we are generating hydroxide ions, and so we're still going to form struvites. We don't recover the struvite as efficiently, and that could be due to formation of hydrogen. The hydrogen could be causing some turbulence on the surface there as well. In other words, you probably have your surface being impeded by both the formation of the solid deposition, but also because you have bubbles on the surface due to hydrogen formation, you don't have surfaces available for conversion as well. And you can see that the efficiency of the negative 1.5 is very low, while the energy consumption is high. This is specific energy consumption in the sense that this is how much energy I am consuming per the amount of phosphorus I recover. So I'm not recovering that much phosphorus, and my voltage is more negative, so it's a high energy consumption, and that's why you see this value here above. So negative 1.5 would not be ideal, which is why we went with negative 1.1, because it gave us the best of all these figures of merit as well, while still generating struvites and removing phosphorus from our system, which again, keep in mind, is our goal. So as far as the experimental analysis was concerned, we looked at the effect of voltage, right? The effect of turbulence in the system. So this is 500 revolutions per minute in terms of one of the conditions, but we looked at different rotation rates, looked at different temperatures, and we looked at different concentrations of calcium as well. And so overall, what we then did is, and this is a plot on the orange bars are the amount of phosphorus recovered. So we want those orange bars to be high because we want to reduce the phosphorus in our solution. And then we want the gray bars, which are represented by these values, to be small, because these gray bars represent the cost of recovery, just one of the costs. There's other costs. So this is operating costs in terms of the electricity. There's other costs in terms of how much does it cost to build your system and things like that. So, but we would want this gray bars to be low, and we want these orange bars to be high. So this analysis was done one factor at a time. So what that means is that we varied the calcium and held the flow conditions, the cell voltage, the temperature, we held those constant, and varied the calcium ion concentration. Then we looked at flow conditions while everything else is held constant. And so this is what you would call one factor at a time analysis. And the optimum conditions there are based on the ratio that I discussed before, which is magnesium is 1.37, the nitrogen is 1, and the phosphorus is 1. So that's the ratio of magnesium to nitrogen to phosphorus. The optimum conditions are when there's no calcium in the system, when I have some sort of intermediate flow conditions, my cell voltage is negative 1.1, and then my temperature is close to ambient temperature. Those are the conditions that optimize my system in terms of cost, right? Lower cost and higher technical performance. Now, let's look at this region where there's calcium. This region is important because in animal waste, you're always going to have calcium present in the system. So while it's great that our technology performed in the absence of calcium, there will be calcium present when you have, for instance, this we focused on swine wastewater here. There'll be calcium present there, and therefore we have to figure out how to better operate the system in the presence of calcium. So what you notice is that as we increase the concentration of calcium, my amount of phosphorus I'm recovering is just significantly decreasing. So there's more phosphorus in the system that just stays in the system, which I'm not removing. So that's not good for what we're trying to do. So while at 210, you can see that the cost is low. Yeah, the cost is low, but you're not doing anything to your solution. So this is really just a moot point. It doesn't matter that the cost is low. It's not operating as we would want. And so the question we needed to answer then is, what is happening here when we have calcium in the system? And to reiterate, these are the plots when we looked at the effect of figures of merit, when we looked at the effect of calcium in the system. So blue, no calcium. Orange here is 72 ppm calcium, which corresponds to about 0.17 in terms of calcium to magnesium ratio. And then this is 210 ppm, which is about 0.5 in terms of the ratio of calcium to magnesium. And so what you see is that when I'm at this 210 ppm, I basically, my current doesn't decrease because I am not forming any struvite on the surface. Now, it's possible you could be forming from some other solid, but if I'm forming some other solid, then I would expect my recovery to be high, right? Maybe I'm forming another solid that doesn't deposit on the surface. Well, that's likely, but the fact that most of my phosphorus still remains in the solution tells me that if I'm forming some other solid, it is not leaving, it's probably redissolving or something like that. So the question was, what is going on here? Why is it that, at significantly high calcium concentration, we get almost no phosphorus removal, but we're still generating current. We still have a high pH at the surface of the electrode, while at 72 parts per million, we get some struvite deposition, and so we get some struvite recovery. But again, is there some way we can figure out what is going on here as well? And so to better do this analysis, and this is the role I played here, is to build what we call a thermodynamic simulation to figure out what are the conditions under which we would get struvite formation in the presence of these other foreign ions. So right now we're just talking about calcium, but there could be competition from other negative species as well. And so what we wanted to do here was control the pH of the solution. And this is important, again, like I said, because the pH determines the forms of these species that I have, really the forms of all these species that are available. And that is going to determine how much struvite I form. And so what we're going to be looking at or measuring is something known as the saturation index, right? Where these concentration values or these activity values are equivalent to what is present in the solution. And this is the equilibrium constant. And so big picture, if the amount of species in the solution are greater than the equilibrium constant, that means that I am saturated and therefore I can, or supersaturated and therefore I can form struvite. On the other hand, if I don't have enough of these species present in the system, then I have no struvite formation because it's undersaturated. And so the concept here was to figure out how we are going to relate the amount of hydroxide we have in the system and include some of these foreign species to figure out whether we're getting struvite or whether we're getting some other species or whether things are remaining in solution. And so this was built in a process simulation software known as Aspen Plus. And really what Aspen Plus can do, it has the thermodynamic parameters that we need to figure out these conditions for saturation, temperature, pressure effects, and things like that. And also you can do kinetics as well. So this was sort of the platform I wanted to build this on. And so in this system, I'm decoupling my nitrogen and phosphorus. So I'm taking, rather than in the experiment, we had ammonium dihydrogen phosphate, but in this system, I'm separating all of those. So magnesium is available as magnesium chloride, nitrogen as ammonium chloride, phosphorus as sodium dihydrogen phosphate, and calcium chloride. And so what you will notice here is that as much as possible, I try to keep the negative ions that are not of interest all the same. So I try to keep those as all chlorides, and then the positive ions that are not of interest, I try to keep as sodium. So that way we don't have this impact of ions, because that will be something that will be interesting to study later on. But for now, let's just look at what we have in our system: nitrogen, phosphorus, magnesium, and calcium, and how that plays a role. And again, keep in mind that the hydroxide ions here are controlling the availability of my phosphates and the availability of my ammonium, all right? And so those are going to be important in determining whether I form struvite. The other thing I want to point out is, in this particular setup, my OH is going to be from sodium hydroxide. Now, that's not going to be what happens in reality. The goal is to connect the electrons or the electricity that's being used in the system to hydroxide. But for now, using caustic soda is a good way to drive that pH. And so we mix our species together, and basically I can control how much hydroxide I add to the system. And that determines what my speciation is, which is going to be important for determining my saturation index. Right, so I'm just skipping. Ben, are there any questions right now? And I wanted to take a break here to see if people had anything so far. Or do you want me to wait till the end? Okay. All right, that's good. All right. And so so the first thing was to look at, and I want to show this plot because you can see how the pH of the system determines the species available, right? So the plots with markers on them, so the circles, the squares, the diamonds, those are my phosphate species. And then the lines without markers are my ammonium or nitrogen-based species. And so what you can see is that there is going to be, based on the pH of my system, I'm going to determine whether I'm going to have ammonium available in the system or whether I'm going to have ammonia, right? And also based on the pH of the system, I'm going to determine whether I have, the hydrogen phosphate, or phosphates. And so this is the way these ions speciate in solution when I have no positive ions to form solids from there. And then, and I had to import or put in the reactions that involve either magnesium or calcium. So both of these are going to be present in there. And what you will see is that I don't just have solid species that can form. So magnesium phosphate is an example of one, and magnesium hydroxide is another. Some of these, some of my magnesium is going to be in solution as well, just based on the fact that you can form some of these aqueous species that are neither solid or just pure magnesium too. And so we had, the methodology here was to look at all the thermodynamic constants for these in terms of the equilibrium constants. And your calcium species also have the same, they sort of mirror each other as well. So you have CaOH plus, you have calcium hydroxide and things like that. And so now once this model was done, and some of the assumptions made here is that you can also have calcium and magnesium bonded to ammonia as well for our main species, but we ignored those for this current iteration, and that's something that can be considered later on. And all of these were considered at 25 degrees Celsius to avoid having to do some regression in terms of temperature. So we just focus this at ambient conditions, 25 degrees Celsius. And so the question was, when I have no calcium in the system and I'm varying my pH, what solids form, right? So going back from my magnesium, my species considered magnesium hydroxide, magnesium phosphate, and of course the chemical of the moment, right, struvite. And so what we see here is that, what we're looking at is where is the potential region for struvite recovery. And so the blue marker here shows the range of where struvite can be generated. And you can see there's a competition here between how much struvite I can form, but that also competes with the formation of magnesium phosphate as well. So some of your phosphorus, remember the ratio here is 1.37 to 1 to 1. So ideally, if we had no magnesium phosphate forming, we could potentially maximize how much struvite you could get out of the system. But what you notice here is that at a pH of 12, we practically get no struvite formation, and that is because around the pH of 12 is when your ammonium basically drops out, and what you have really is ammonia in the system, which in reality, in a real life experiment, if you drive your pH that far, you're going to form NH3, which is going to basically, ammonia is volatile, so you will just leave your solution and you're basically using your nitrogen to air. So you're not really recovering nitrogen in that manner. But there is a potential window where I would have struvite formation. So now once calcium is introduced into the system, and so this is calcium at 0.17, which correlates to our 72 ppm. And this calcium to magnesium at 0.49, which is correlates to our 210 ppm. And so you can see there's a competition there between, you can see that your potential struvite recovery decreases. The range which we had here was between, like I said, roughly 6.8 and 12. Let's just say between 6.5 and 12. And you can see that window is going to shrink based on the presence of calcium in your system. And the more calcium you get, the more that window is going to shrink, because you can see that the formation of calcium phosphate basically is going to, the potential for calcium deposition or calcium phosphate formation occurs before you're in a region where you can actually generate or form struvite. Now I want people to keep in mind that this doesn't include, or we haven't looked at kinetics analysis here. So it's very possible that the rate of formation of struvite is faster than your rate of formation of calcium phosphate. And therefore, even though thermodynamically you can form calcium phosphate earlier, you might form more struvite than possible. What this will give us, though, is sort of a window where we would want to hypothetically operate. We know definitely that if your pH is lower than this, under these conditions of 1.37 to 1 to 1, you're not going to get any, you won't get any struvite, which is what thermodynamically is not feasible to form struvite there. Okay. So then based on this, the goal was now just let's look at different calcium ratios between 0 and 0.5 And that is how we generate it. So what you're looking at here is a heat map where you have pH on the x-axis and calcium to magnesium ratio on the y-axis. And basically you can see where the potential for struvite formation is. Again, you have this cutoff at 12 because of the elimination of ammonium from your system, because you mostly form ammonia. But what this is telling us is that once we have calcium in our system, our operating window has to change because our pH window is different. Our pH window shrinks. And so now recall, how are we controlling our pH in this system? Well, we're controlling our pH by the voltage we apply, right? At lower voltages, you're reducing oxygen and generating hydroxide ions, and then at higher voltages, we are driving water reduction to hydrogen. And so we're controlling our OH by the potential we apply. And so remember I said that this work was done, one factor at a time. And so what this possibly is telling us is that for us to actually get any significance or to really get struvite forming in the system, is potentially possible that we need to drive our OH concentrations to higher pHs, which means we would need to raise our voltages to make that work. Now, like I said before, you're going to be competing. Now, this is under when we have calcium. So the previous picture I showed you, when there was no calcium in the system, when my voltage was really high, my recovery went down. But the goal is now, if we have calcium in the system, it would make sense to perhaps operate at that negative 1.5 volts. The other thing is the calcium species that are being formed, and this is based on literature. The calcium species you would form are relatively unstable. So even though we're having this competition and we're forming calcium and calcium is forming these solids, the solids we're forming appear to be solids that you form them and then they redissolve. So they're not really, so you're forming a solid material, but that solid material is redissolving in your system because of the fact that you have most of your phosphorus not being recovered as well. So this is, again, giving us just a window into how we would want to operate this system. We would want to drive our voltage probably to likely higher voltages. And then we need to consider what happens. How do we may need to raise our magnesium in our system as well. And the other thing to keep in mind is that, yes, we want to drive our pHs to higher values, but we also want to consider the fact that we still want to have nitrogen in the system. So if we drive our pH too high, we would basically eliminate the ammonium from the system, and then we'll have issues. And so just to summarize here, we, based on, and again, a lot of these details are in that paper, but I just wanted to give you a flavor of what the goal here is. We're able to couple the thermodynamic simulation to explain the experimental results for us. And ideally, what you would want to do is do the thermodynamic simulations first, and then go into the lab and do experimental work to figure out what operational window you want to. So we did this a little bit backward, but the theory provided insight, and so future work we're going to do will look at some of these effects prior to us actually going into the lab. And really, that is the work that I'm going to be carrying on starting as an assistant professor. So part of the work we still need to do, remember I said this is one factor at a time analysis. Well, what the question is, what happens if I have, calcium in my system and I'm operating at, negative 1.5, a potential of negative 1.5 for the cathode? Do these conditions change, right? And that's where the assessment of multifactorial effects will come into play. And we're actually starting to do some of this work already. So I'll introduce you to some of the people in my group in a sec. But some of the work that we're currently carrying out is looking at the effect of temperature on the formation of these species. So I mentioned before that what you saw was all at 25 degrees Celsius. But now if I look at pH on my x-axis and look at the effect of temperature, we can see, and now we have more calcium species there as well. Because the other thing to do is we limited ourselves to 2 solid species per positive ion. So we had calcium phosphate, calcium hydroxide, and we had magnesium phosphate, magnesium hydroxide, and of course struvite. But I tell you, if you were to just type into Google right now calcium phosphates, you will see there's probably about 7 to 10 different forms of calcium phosphates, similar to magnesium as well. So that's work that we need to do into incorporating some of these other species into our thermodynamic simulation, and then consider which ones are probably not kinetically feasible, so we can just focus on the ones that are. But what you're seeing here is that when I have these other species here, well, now I have competition for phosphate, not just due to this calcium hydrogen phosphate, but I also have competition due to what you call amorphous calcium phosphates at the higher pHs as well. So there are going to be interlocking factors aside from these temperature effects and pH effects as well. So like anything else, you to get any work done, you need someone to sponsor that work, right? So in this case, it's the Ohio Water Development Authority that's facilitated this project from the get-go. So those previous results were from a 2016 grant, and they funded another project to go from 2019 to 2021. So we're grateful for their support. And also my colleagues who started this work at the Institute, who were Zineb, Dr. Zineb and Dr. Jason. And then these are my two students, starting off as a faculty member at Ohio University. Garrett is an undergraduate who is going to be looking and focusing on the analysis for the thermodynamic simulation. And he's actually he's doing interviews, he's going to be doing interviews in April for grad school. So he's a really good, great candidate. And then Tunde is my graduate student, PhD student, who is going to be working on the experimental work. And so they will be the ones doing the heavy lifting, and with a little bit of my help on that future work that I mentioned. And so with that, I will take any questions. These are some of the ways in which you can reach me. That's our website. That's my email. And of course, I found Ben and I found each other through Twitter. So I figured it was appropriate to put my Twitter handle as well.
Absolutely.
I'll take any questions now.
Awesome. Thank you very much for a fascinating talk. So we have a good number of questions already. And I'll say if there's anybody, I saw a couple of new names. So if you're new and you would like to ask some questions, just make sure you hit follow. You'll be able to type in the chat and you can ask away. And so I'll just go ahead and jump straight into it. So I have a number of these are mine, but because I get really interested and start asking questions so I don't forget them.
No
all right, so the first one I have here is what kind of time delay is there between the formation of the hydroxide ions and the formation of the struvite? Do you have like a ballpark idea?
So I think one of the ways to look at that, here we go. Okay, so you can, if we were to, and I can't zoom in here, maybe I actually know what I could probably do that. Okay, remember I said that the removal or the reduction in current here is related to the position on the electrode.
Yeah.
So we can get a sense of when that starts to happen, right? Because you can see when your current actually starts to decrease on this time frame. So it happens relatively quickly. I would say, I don't want to say within. I don't want to say probably within minutes.
Okay.
Whether that's a minute or 2 minutes is I can't quite quantify that. And
sure,
I will say this: the other thing to keep in mind is that because we have this impeller which is continually rotating, then that helps with any mass transfer limitations we might consider too. So that helps to move that along relatively quickly.
Okay, that makes sense. So what causes when you had a slide a little bit earlier, slide 8, I actually remembered to write it down. So at minus 1.1, there's the really steep drop, and minus 0.8 is kind of a gradual decrease.
Right.
At minus 0.6, you get sort of an initial steep drop that matches the minus 1.1, and then it levels off. Do you know why you get that sudden drop at minus 0.6 that's not there at minus 0.8?
Oh, you're saying how quickly this happens?
Yeah.
In this analysis, because I think in this particular case, right, we didn't quantify the statistics of how often this happens. I mean, some of this behavior you see are relatively pretty repeatable.
Yeah.
Now this was done in an RC configuration, so let me think. Because at those voltages, like I mentioned, what we have there is mostly oxygen reduction, and how much there's a finite amount of oxygen, so it's not like negative 1.1 where you start to get some water reduction, right? And so there's a finite amount of oxygen present in that system. Now you'd probably expect that at negative 0.8, where you'd have more oxygen conversion, this would drop off quickly, but I'm not And that's something to think about.
Okay. Yeah, I was just curious because I mean, like you said, it may just be the conditions just happen to be just right for it to do that sudden drop off and then level out. So I was just curious. So from Dr. Boyka, we have a question. Oh, so he was wondering why not just add sodium hydroxide and do the generation of the OH electrochemically?
Cost. Yeah. So if you
there you go.
Yeah, like the cost of, I think if we compare the cost of electrons to the cost of, and I did this quantification somewhere.
Mm-hmm.
The cost of hydroxides from electrons and the cost of hydroxides from sodium hydroxide in this configuration were probably, sodium hydroxide was probably 4 times that.
Okay.
And so that's why a lot of times the people who are doing this at scale are not using, they might use a little bit of sodium hydroxide, but they're also using like magnesium hydroxide, which is, and things like magnesium oxide actually, because that magnesium oxide can drive your pH and is cheaper than sodium hydroxide, but that's the cost associated with that.
That's I actually would not. I didn't realize that. That's interesting. Okay. Okay. So. Okay. Here, this is what I was wondering. Could you purposefully form struvite in such a way that it actually doesn't stick to the electrode? So you noted that as you rotated more quickly, it created more turbulence. At least some of the struvite basically formed but fell off. Can you envision a way where you could do that purposefully, so that so instead of having to like stop, pull out the electrode, either put a new one in or scrape off the old one, could you do that on purpose?
Yeah. So one of the ways you could do that is the flow conditions, and really, the interesting thing is, and you will see this actually happens. So this struvite formation happens at wastewater plants, right? When you have this excess phosphorus as well. And so struvite actually has very strong interactions with steel, right? And so one way to do it, and again, that's the balance between how much it's going to cost versus how well it's going to perform, might be to use a metal, an electrode that is still good at oxygen reduction and water reduction, but doesn't, attract struvite as much. But if we still with our stainless steel, then one thing to do might be to, increase flow rate or introduce something like sonication or something like that can, remove this struvite from the surface. But that's a crucial part of figuring out how to make sure that this technology is scalable, because if we don't deal with that, then, we basically just have a batch process, which is not, economically that's not going to work.
Okay, so yeah. I guess kind of also it's interesting you said scalability, because that's actually the next question is, what is the scalability? And I guess I'll piggyback on that and say or ask, could you, given that there seems like there's at least a minute or two delay in the process that you have set up now, would it be possible to have like a flow through setup where you do this formation of the OH, but by the time it gets away, by the time the struvite is forming, you have it separated from the electrode such that the electrode itself can still keep going without that deposition.
Yeah, that's a really great point, and I think one of the goal is, like most of this, right? We're going to start with this batch process first, understand our parameters, and then
absolutely,
right, and then we'll take continuous step. But that would be one of the ways. You're right. That delay is something we can take advantage of and actually do a flow through system, and then figure out how we can. And even in the continuous process as well, right? You could drive your flow rate so high that it would basically just share off some of that struvite. There's some electrochemical tricks you could play, right, by doing things
absolutely.
Like step up, step down type voltage, and see how that might make that work. But again, those are like sort of. Then you're getting into, okay, well, now that's where the art comes into play, right? It's not,
absolutely.
It's more art than science.
Absolutely.
But that's a really good point. That's something that we would like to. So I think our next step really is, once we get this multi-factor analysis, we have a good sense of how these different parameters interplay. We can then start to let's move from a batch process to an actual continuous process that takes advantage of some of these key characteristics of our system. Okay,
so I guess so that kind of goes into the scalability question, and Dr. Boyka also asked about the economic feasibility. So do you think that that's kind of the step that needs to happen to push this app in terms of an actual application? Yeah. Is that kind of the step that needs to happen to bring it to that point?
yeah, definitely going from a batch to a continuous process is a key hurdle that we have to overcome. Because
Sure.
otherwise, it's like, if and again, we're looking at this. If we're considering focusing this on agriculture, or even if it was a wastewater treatment plant, those are not. You wouldn't call those like high. What's the word I'm looking for? High margin type markets, right? They would.
Sure.
Those kinds of markets want cheap technologies. They don't want something that either it's going to be something that's going to give them a headache in terms of operation, or it's like something they have to like start and stop all the time.
Right.
So that's why I mentioned that. Yes, like cheap electrodes and cheap components are going to be useful, but then a way in which you can have something that stands alone and can sort of like not self-operation, but like can sort of operate without too much disruption to their actual bread and butter work would be critical. So yes, making it a continuous process would be critical to making this actually successful.
Okay. So I guess, and I apologize if you said this at the beginning, what's the ballpark amount of waste that one of these would need to be able to handle? Like, are we talking, like hundreds of tons, thousands of tons? I mean, obviously it depends on the size of the farm or the whatever the animal facility or whatever it is, but
yeah. So I mean, let me think about what I remember, which is the most recent calculation I did was looking at wastewater treatment plants.
Mm-hmm.
And so I think so these vary in terms of how much water they treat, but maybe a standard one is like maybe 10 million gallons per day, right?
Okay.
And so the stream of right, but that 10 million gallons per day, the phosphorus or nitrogen levels in those are usually really small, like maybe 4 ppm or like really low levels.
Okay.
But the part that really needs treatment is the part after the digester. Mm-hmm. And the digester is usually like, maybe say the digester is usually maybe 10%. Sorry, not 10%. That's a high number. Usually anywhere from like 0.1 to like 1% of the overall flow. So let's say now you're like instead of 10 million gallons, you're like 100,000 gallons, right?
Okay.
And then your phosphorus is like maybe 500 ppm of that.
Okay.
So and I have a calculator here, but I'm like just thinking like those are some of the parameters that you think of. So 500 ppm or 500 milligrams per liter of your phosphorus, and then you have maybe 100,000 gallon per day type stream, and then you can see how that might work. So I don't remember those numbers, but that'll be an example of a standard one.
Okay.
For an animal farm, you would expect higher concentrations, but the volumes would be lower.
Okay, that's good to know. I mean, that's a much more, that's a doable thing.
Yeah.
Okay.
It's like, I mean, something like that would be like something that I'm sure, because I think Clemson is your in South Carolina, Clemson is the land grant, right?
Mm-hmm.
Yeah, so they would have like some of the numbers that I was even using in terms of like ratios and stuff like that I got from their website. So like, so some of those would be those are some examples of numbers.
That makes sense. Yeah. Okay, so on slide 9, when you're talking about the product efficiency formation, are you defining product as just the struvite, or are you talking about the formation of OH as your? Oh. Okay, so yeah, so are you talking
about this one?
Yeah. Mm-hmm.
Okay.
So yeah, so what do you include in products there? Is it just the struvite?
Yeah. So and that's why I mentioned that this is sort of like an indirect process, right? So the way we're accounting for this is how many electrons do I need how many electrons are generating OH, and then how much OH is generating phosphate, which is a 1-to-1 ratio between phosphate and struvite. Okay. And so in this case, we can quantify that because we're in the synthetic wastewater. We know that we start with it's basically ammonium dihydrogen phosphate. So it's H2PO4 minus, right? And so let me go back to here. Yeah, here we go. So if I'm starting with H2PO4 minus, I just need 2 hydroxide ions to get it to PO4.
Yeah.
So the way that efficiency was calculated is how much, based on my current, I can figure out, I know how much product I formed. I can back calculate that, how many electrons I need based on the OH. And then that's why some of that number is really low. Now, there's some things we have to consider there because I'm making an implicit assumption that all my OH goes to taking my H2PO4 to form phosphate.
Right.
So it's sort of like a high value, right? So And so even though we're calculating this corrective efficiency because it's something that you want to know electrochemically, it's not like what you would typically do in a typical electrochemical system. So I think and that was a question we got from one of the reviewers. So I said, well, this is how we're calculating that. This is what it really means. We're taking the electrons to OH to PO4, and that's how we're calculating that efficiency.
Okay, I mean that makes sense. I think it's a reasonable way to do it, for sure.
Yeah, we have to have something, right, and then we can make improvements later on.
Yeah, exactly, and especially, I mean, as long as you define exactly what it is that you're doing, I see no problem with doing it that way because, like I said, I think it's a reasonable way to at least try and correlate something there.
Right.
Okay, so at minus 0.8, it seems like you get an initial drop and then it sort of levels off. Do you have an idea for why? Why you don't? I mean, it seems like you form at least some struvite, and maybe even it's starting to slowly go up over time. But it seems like why does it not seem to really continue to form the struvite the same way that the higher potentials do?
Right. So I think, and this is something we're just discussing recently. So and this is an open system, right? And so one of the things we haven't quantified yet is, okay, what is the rate at which oxygen is being reduced? And then what is the rate at which, so at negative 0.8 is mostly oxygen reduction. We don't have any, there's no hydrogen generation there at all. So it's mostly this reaction here. Okay. And so, so it's plausible that what happens here initially, we generate struvite, and really what is going on is that we may have consumed quite a bit of our oxygen in the system. And so we get that initial struvite deposition, but now we're not dissolving enough oxygen quickly in the system because we're not pumping oxygen. We're not pumping oxygen into the system. Okay. So one of the ways I might think of that is it possible that we've limited how much oxygen we can actually convert in that system, and therefore that initial struvite is just due to that initial oxygen that was present in the system. And what we know is that, right? What we know is that for oxygen dissolution, the solubility of oxygen decreases not just due to temperature, but it decreases based on concentration of salts as well. Sure. So like pure water might have a certain solubility, but now that we have these ionic species present there, we're going to have sort of a lower solubility of oxygen. So that's going to limit how much oxygen we have available. Okay. As well. So what we figure is that at this, there's some balance there between us. We might have at a higher potential, we might have some water reduction, but again, we're not, we don't want that to be the goal either. We don't want to just be generating hydrogen because we know that's going to compete with our surface and things like that. So there's sort of a sweet spot there.
So maybe this is it's either a really dumb question or a big brain one. No. Could you bubble? I mean, not even just pure oxygen, but like bubble air in during the reaction.
Yeah, you could. Yes.
Okay.
Good.
I mean, I know this is sort of just kind of a educated guess kind of thing, but do you expect that would push your phosphate removal up? I mean, let's assume that like the bubbles don't knock it off of the electrode, that kind of thing.
Yeah, I think we would expect it to go up because again, we are and so one of the ways we can control this better is probably just we'll seal it off, and so we know we can quantify how much oxygen we're sending in there, and then we say, all right, so this is sealed off, we are sending oxygen in, now let's look at how that reacts. But then again, something that we should get a good sense of, and it's sort of an open-ended question right now, is at what rate are we going to be dissolving oxygen? What are the kinetics of oxygen dissolution in our system?
Sure.
And then how does that compare to the kinetics of oxygen conversion, and do those balance each other out or not?
Okay, that would be an important question for sure.
Yeah.
Okay. So Dr. Boyka had a question about the chemistry of the calcium 2+. You kind of touched on this with the thermodynamics work, but yeah, he was especially curious about the effect of the 2+, the calcium 2+ on the struvite formation.
Right. So there are. Well, first off, when you have calcium in the system, the calcium is going to compete with phosphate. Okay, or it's going to compete with your magnesium for phosphate. As you can see in this table, basically for every magnesium There's no calcium-based struvite, but for every magnesium species you have here, there's a corresponding calcium species as well. And again, this is limited in terms of the solids that we have quantified, right? Based on our thermodynamics. There are still tons of other phosphates that we're going to be including in the system.
Okay.
And so what we foresee is that your calcium, first of all, the onset formation of calcium phosphate, and this calcium phosphate was just tricalcium. So again, keep in mind that there are different forms of this. And so that the onset or formation for that, or the possible potential formation of that is happens, at a much lower pH than it does for magnesium. Now, I haven't seen any data yet on this. It's also very possible that the kinetics of this will be different from that for struvite as well.
Okay.
I'm not sure which one is faster or slower, but when we were doing a literature review, we discovered that, in terms of calcium species formation, the most stable one is the one known as hydroxyapatite. So it's like Ca, it has phosphate in it, it has hydroxide in it.
Oh, okay.
And some of these calcium phosphate species are precursors for that. But there is an effect of calcium species nucleation based on the amount of magnesium you have available. So there was a group in New York that did, and they were doing this work for for dental applications, because calcium phosphates are used in dental applications quite a bit. And so they're looking at the effect of having magnesium present in your solution and how you can nucleate calcium species as well. So their summary there was that when you have these competing species, you might form calcium phosphate, but that calcium phosphate is unstable, so it's going to redissolve. And so what we're thinking is happening is that getting through, you may get some deposition on the surface, but quite a bit of your calcium is basically reacting with your phosphate and almost like redissolving. So it seems like a continuous reaction. The other thing to keep in mind, and this is important as well, is at the anode, we're oxidizing water. Okay. And so you're starting to get some environments that are likely to redissolve some of these species that form as well, because the calcium phosphate doesn't form on the electrode. It's possible that it probably just forms and crashes out of the solution. And so we were thinking, okay, well, if we're forming calcium phosphate and it's a solid species, well, we should be recovering it from the solution, but it's not, right? You can see that tells us or leads us to think that we might be forming the solid, but it's basically redissolving in our system, because it doesn't explain what it is now. We may be able to negate that, like I said, because if we look at this window here, if we drive if we increase our voltage so that we have more OH and our pH is higher, then we will get to a region where we can sort of negate some of those effects. So and what you're doing there is you're creating a region for supersaturation for struvite. Okay. So we haven't figured that out yet, because in this analysis, when we varied calcium, we held the voltage constant, right? So the question is, if we went to negative 1.3 or negative 1.5, would we get more deposition due to the fact that you've changed your potential, or you've changed your sort of your thermodynamic possibilities, and so now you have in a region where you can form that struvite. So that's why I said, this was good in terms of the fact that it told us the parameters, but now we need to figure out if there are crossing effects of these different parameters on each other, and that's where we want to go with the work.
Okay. Do you have any plans to look at a separated cell, so like a membrane separator, or even just like an H cell with fruited glass or something like that?
Right.
Would that hold any attraction?
Yeah, that I think that is of interest. The question then becomes again, you introduce a membrane, you have another over potential to deal with. So it will be good to do that though, because if you're forming hydrogen, right, then an H cell will make sense, because now you can say, okay, I'm forming hydrogen, I can collect that hydrogen and basically maybe sell that or whatever, right? So there are advantages to introduce this into separation. And there's a group, I believe, in Arkansas I think they've looked at both configurations. I think they looked at both an H cell configuration and just a regular undivided cell configuration
Mm-hmm.
as well. And so they're sort of like the people that are also doing this work in this space. Yeah.
Cool. All right. If there are any further questions, go ahead and get them in now. Yeah, this was fascinating.
Those are really good questions. You know, made me think, but it's good because I mean, so AICHE, the conference is next week.
Oh yeah.
And I have to, so we'll present some of this work there, and so I have to like, and because it's virtual this year, we have to record our presentations beforehand and
Oh yeah.
And upload it, and so
Mm-hmm.
so this was a good. Now, obviously, I'm limited to 15 minutes there, so I'm not going to
Sure. Sure. Yeah.
a lot less superfluous, but
Hey, no, like I said, this that's why I like that we don't have to put a limit on it, because I feel like we got to go into a lot more detail, which is like that's the fun part for me. This is really cool. Like I said, I was peripherally aware of wastewater remediation work, but I really didn't know a lot about it, and I feel like I know a whole lot more about it now, and it's a really fascinating field.
And if you're really interested, so I told you my advisor works in this space, so her work is more on the nitrogen side, so she's looking at ammonia, urea, and things like that, and that's kind of like what started me on this journey on like looking at electrochemistry and wastewater. And so it's So there's the nitrogen people, and then there's the phosphorus people, and then that's sort of like the dividing line in terms of figuring out how to treat water with these nutrients. But it's key. It's key.
Oh, absolutely, yeah. It's a big issue. It's good. I'm glad people are working on it, because Lord knows we need to. Oh well, I think that's all the questions we've got tonight. So unless anybody has any last minute ones, I will go ahead and say thank you to you, Dami. Like I said, fantastic presentation, super interesting, and I'm glad to hear that. And I will thank everybody for coming out, and I hope to see you all next week. We have another great talk lined up, and let me forget who it is. Let me pull that up real quick if I can. Oh yeah, there's Dr. Troy Olivio from Nicholls State. He's going to be talking about course creation, especially focusing on the current generations coming through college, and also some of the things that he wished he had known as a new PI at a primarily undergraduate institution. So that should be a great talk. I hope everybody will come out, and I would say thank you again to Dami. Like I said, fantastic talk. And
thank you so much. Thank you, everybody. I hope you got some useful information.
Oh yeah, you got a lot of compliments in the chat here. They really enjoyed the talk. So yeah, I'd say mission accomplished on that.
I appreciate it.
And if I could get you to hang around for just a second, Dami, we'll talk real quick. And with that, thanks everybody. Have a great weekend. I'll see you next week.